Welcome to IPMWORKS e-learning modules. My name is Ellie Dearlove from ADAS, and this video is part of Module 5 Invertebrate IPM. I will present Chapter 5.5 Biocontrol of Invertebrate Pests. The aim of this chapter is to summarise the use of biocontrol technologies to manage invertebrate pests. We will cover what are biocontrol technologies, classical, conservation and augmentative biocontrol, predators, parasitoids and pathogens, other natural substances used as biopesticides, and finally, other beneficial invertebrates as part of the agroecosystem. The integrated pest management strategy of biocontrol involves using natural resources to control pest populations. There are different types of biocontrol technologies available, and these include invertebrate biocontrol agents. These are natural enemies such as insect, mite and nematode species that provide control of pest populations through predation or parasitism. Microbials, on the other hand, are based on microorganisms. Semiochemicals are substances emitted by plants, animals and other organisms used for intra- and interspecies communication, and they have a target-specific and non-toxic mode of action. Natural substances consist of one or more components that are either sourced from nature or are nature identical if synthesized. We often group these technologies further. Predators, parasitoids and pathogens are often referred to as natural enemies. We can also group microbials, semiochemicals and other natural substances under the term biopesticide, and we will cover all of these throughout this chapter. Natural enemies and biopesticides are often more targeted towards the pest and disease in question, and their application helps deliver economically viable crop yields when used as a component of integrated pest management programs. While not a like-for-like substitute, biocontrol can help reduce the use of chemical pesticides and provide an important tool in resistance management. Several biocontrol technologies can be used in classical, augmentative, or conservation biological control. There are different types of biological control: classical, conservation, and augmentative. Classical biological control is the introduction of a non-native natural enemy for the permanent control of a pest. This form of control is mainly used for the control of invasive species, although some non-native species can be released under license as part of an IPM program in protected cropping environments. Very little classical biological control is applied in IPM due to the risks associated with the introduction of non-native predators. And so we will focus on conservation and augmentative biological control. Conservation biological control involves manipulating the habitat in and around the crop to increase the population of native biocontrol agents. For example, flowering field margins managed around arable crops can provide resources for ladybirds, and in protected crops, banker plants can be used to host additional food sources, providing a consistent source of food for generalist beneficials. Augmentative biological control is the release of additional numbers of a natural enemy when too few are present to control a pest effectively. Many biocontrol agents are produced commercially and can be released in great numbers, as shown in the central image. The surrounding images show various predators which can be used for biological control of thrips, whitefly, aphids, and moths. Conservation and augmentative biological control can be combined so that when additional natural enemies are introduced, their populations can self-sustain using resources provided within and around the crop. For microbial control agents, such as predators, supplementary food can also be purchased to increase numbers of natural and released organisms and aid establishment in the crop. For example, moth eggs can be used to feed the predatory bug Macrolophus pygmaeus. Conservation biological control is most commonly used in outdoor crops, whilst augmentative control is commonly used in protected crops. Biological control can have a significant impact on pest numbers within a crop. This figure is adapted from work looking at the impact of natural enemies on aphids in cereal crops, and it shows the change in aphid abundance in response to the number of natural enemies within a 2-week period. Here we can see that in the absence of natural enemies, at green point 1, there is a relatively large increase in aphid abundance. As the number of natural enemies increases, the increase in aphid numbers is slowed until at point 2, there is a point at which enough natural enemies were present to reduce the number of aphids. It's important to consider here that, as discussed in chapter 5.1, the crop can tolerate some pests without impact on yield, and so the aim is not to eliminate the pest, but to manage their populations to a level that does not exceed economic thresholds. It's also important to recognise that while increasing the number of natural enemies is important, the main benefit is seen in ensuring that natural enemies are present. It is in the absence of natural enemies that pest populations become very large very quickly. There is sufficient evidence from across Europe, in all crops, to be assured that natural enemies of crop pests are making crucial contribution to IPM, and crop management decisions should account for these important invertebrates. This can be achieved by firstly ensuring there are resources available for them in and around the crop, and secondly, taking a holistic approach to ensure that management targeting one aspect of crop production does not greatly reduce populations of beneficial invertebrates. Conservation biological control addresses a core challenge in making the most of natural enemies, which is that the predators and parasitoids of crop pests require a different range of resources than that of the pest, and these resources are often not available within the crops. For example, if we consider hoverflies as aphid predators, these predators are common across Europe, and their larvae are highly effective predators of many species of pest aphids. The adult lays eggs on plants near aphid colonies, and the larvae consume hundreds of aphids before pupating. When the adult emerges, it does not eat aphids, instead it feeds on pollen and nectar, and they must therefore migrate to flowers to feed. If flowers are in short supply, this limits the population. This split in food source between adult and larval stages is common in insects. In addition to the need for floral resources, natural enemies also need access to alternative prey when pest populations are not high in abundance. Winter habitats are also essential, as many do not survive well in overwintering crop habitats. As demonstrated in this figure, the resources needed also changes during the year, showing the need for year-round management to make the most out of these beneficial insects. Natural enemies make an important contribution to pest management, but they too need to be provided for within production systems. Whether in outdoor or protected crops, it's important to ensure that they have the resources they need across the whole life cycle. Care must be taken to ensure provision of these resources does not itself promote other pests. Each natural enemy has a different range of resource requirements, and so it's important to identify which natural enemies are found around the crop and target resources to their needs. For more information, visit EcoStack, an EU-funded project that develops and supports ecologically, economically and socially sustainable crop production via stacking and protection of functional biodiversity. We will now give an overview of a few other natural enemies used in augmentative control, mostly in protected crops. As well as promoting naturally occurring populations of aphid predators in outdoor crops, aphid predators are widely used in IPM programs for protected crops. These images show a predatory midge larva, the lacewing larva, a ladybird larva, and a hoverfly larva. And these predators feed on most aphid species and can be bought commercially to introduce to the cropping system. Parasitoid wasps kill pests by laying their eggs within or next to their chosen host. The developing larva kills its host as it develops into an adult, and this is what determines a parasitoid from a parasite. There are around 9,000 species of parasitoid wasps, and they tend to be specialists, targeting a narrow species range of hosts. Parasitoids are used in IPM programs in protected crops, and they can also occur naturally both under protection and outdoors. It's important to use the correct parasitoid species for the pest species infesting the crop due to their specificity. Several parasitoid species are available, and mixtures of parasitoid species are available for crops attacked by several pest species. Parasitoid wasps are commonly used to control aphid populations due to the wasp's ability to find aphids at low densities. First, the parasitoid wasp female locates a host and uses her ovipositor to lay an egg inside the aphid. The parasitoid larva hatches and develops through several larval instars inside the aphid, killing the host. The parasitoid larva spins a cocoon in which to metamorphosize into an adult. This changes the appearance of the aphid into what we refer to as a mummy. Different parasitoid wasp species produce different aphid mummies, as shown in figure 3A, B and C. The fully developed adult wasp emerges from the mummy by cutting a circular hole. In order to keep aphid numbers low, weekly preventative parasitoid releases can be conducted as soon as temperatures are suitable. Increase the release rate to a curative rate when aphids are present. Then, monitor the plants regularly to check that aphid mummies are developing and that the percentage parasitism is high. Increase the release rate if needed, or start releases of aphid predators to supplement control by parasitoids. One problem can be that the primary parasitoid inside the aphid mummy can be attacked by a hyperparasitoid. The parasitoid adult emergence hole is smooth and round in normal instances, whereas the hyperparasitoid emergence hole is ragged. Hyperparasitoids can threaten biological control of aphids, so if these unusual emergence holes are noticed within the crop, it's recommended to use predators as a supplement to the IPM program. Entomopathogenic nematodes are another type of natural enemy that can be used in biocontrol. They occur naturally in the soil and they can be purchased as a commercial product for application through augmentative control. The third larval instar nematode is known as an infective juvenile. They are not able to feed and they must find a host in which to mature. The nematodes enter their host through natural openings such as the mouth or anus, and they release bacteria which produce toxins to kill the insect and digest its bodily contents. The insect dies and the infective juveniles use the digested body contents to develop into the fourth instar larvae. The fourth instar larvae sexually mature into male and female adults and reproduce sexually. The female produces eggs which develop inside her body and kill her, as shown in the video. If there are enough resources in the insect, several generations of nematodes can be produced. When the resources run out, the third instar, infective juveniles, emerge from the insect body, as they are the only life stage that's able to survive outside of the host. Nematodes can be bought and suspended in water to be applied to the soil and target species that are within the soil or have a part of their life cycle within the soil. Beneficial invertebrates are effective at reducing pest populations. However, microbials also play an important role as natural enemies. Microbial natural enemies are entomopathogens. These are pathogens that infect and kill insects. These are including but not limited to bacteria, fungi, protozoans, viruses, viroids, peptides, mycoplasmas. And they may also include entire microorganisms, living or dead cells, any associated microbial metabolites, fermentation materials, or cell fragments. Most of these are target-specific, so they have little to no non-target effects. And entomopathogens can be used in classical biological control or conservation biological control, where we manipulate the habitat to suit their requirements. However, many entomopathogens can be mass-produced and applied using the same or similar equipment as that used for chemical pesticides in augmentative control. In these instances, we call them biopesticides. In addition to other natural enemies, biopesticides are an effective tool as part of an IPM strategy. The term biopesticide groups microbials, semiochemicals, and other natural substances together when used as a plant protection product. The microbials have the capacity to kill pests or outcompete and prevent diseases. Semiochemicals are message-bearing compounds produced by an animal or a plant that can be used to change and disrupt a pest's normal behaviour. These are often used for mating disruption or monitoring. Finally, natural substances are naturally produced or nature-identical compounds obtained from plants, minerals, and animals that have antimicrobial, insecticidal, or pest repellent activity. The most common type of semiochemical used for invertebrate control are pheromones. These are often used in traps to monitor pest numbers and can also be used for mating disruption. A case study within IPMWORKS conducted in Slovenia on 3 different locations has shown the success of mating disruption using pheromones. The European grapevine moth has 2 to 3 generations per year depending on its location. Caterpillars damage flower clusters and grapes by feeding. In May and June, the first generation larvae web and feed on the flower clusters. The second generation larvae feed on the green berries. And young larvae penetrate the berry and hollow them out, leaving the skin and seeds. The third generation larvae cause the greatest damage by webbing and feeding inside the berries and within bunches, which become contaminated with frass. They bore into the berry and they feed from the inside, so there is little intervention that can be done once the insect has started causing damage. But in this case study, high concentrations of a synthetic sexual pheromone, analogous to the female attracting pheromone, successfully caused male confusion in locating females. In 3 vineyards, percentage damaged bunch of grapes were reduced to 5% or less when implementing this approach. This is a residue-free control method and a good resistance management technique due to the reduced applications of pesticides required. It's important to consider that the pheromone is only used for adult mating disruption, so the timing for use each generation is essential, and the monitoring of the emergence of adults is required. Rock powder is a natural substance that can be used alone or in combination with copper and aldehydes against olive fly. The powder is mixed with water and applied across the entire canopy of olive trees. The fine coating on the tree and fruits makes the olive difficult to locate by the fly by altering the colour, texture and smell. The white coloration is also useful at retaining water and preventing excessive heat in the crop. Within the IPMWORKS olive case study, plants were treated with zeolite and recorded a lower fly attack than untreated plants which had 50% affected fruit. Rock powder is easily washed away with rain and does not affect the market value of the crop. There are several general considerations which should be made across all biological control technologies. It's very important to plan your biological control program as part of IPM. As with conventional pesticides, for a biological control to be effective, farmers need an exact identification of the pest and to understand its biology and ecology to match. We also need to consider the ecology of the invertebrate pest in relation to the biological, ensuring that the timing of application is suitable and providing additional resources for the biological if required. When applying a chemical pesticide to outdoor crops, farmers must also consider wind speed, rain and temperature, growth stage of the crop and the pest. Similarly, these conditions must be considered before applying a biological. This is especially true if we're using a living organism. The required conditions will differ according to the biological being used, but as a general rule, extreme temperature conditions are not suitable for most biological living organisms. Microbial biopesticides often have humidity requirements and UV exposure can be damaging. We should monitor and regularly adapt our program if needed. If other control agents are required, choose these carefully to ensure their compatibility with your biocontrols, and understand that biocontrol may not eradicate the pests, but with careful management they should be able to keep them under control. There may also be adjustments required for the storage, application and timing of application depending on the biological technology used. In the next few slides, we will cover a few specific examples of these considerations when using different biological technologies. As mentioned previously, understanding life cycles of the pests and predators in your crop is essential. For example, ladybirds and hoverflies are well-known predators of aphids. Both the larvae and adults of ladybirds will feed on adults, but only the larvae of hoverflies will feed on aphids. The adults feed on pollen, nectar and honeydew, and therefore it's important that they have access to flowering plants in order to complete their life cycle. Some predators are known as generalists because they can attack many different species of invertebrate, or their diet may also consist of plants and fungi. Specialists, on the other hand, attack a narrow range of species within the same genus or class. In this table, we can see that different species of predatory mite will consume all or just some of the 3 pests listed. And so it's important to correctly identify your pest and select an appropriate predator. Different predatory mite species also have different requirements for environmental conditions. Most predatory mite species are vulnerable to low relative humidity and will tend to stay within the microclimate of the crop canopy where relative humidity is higher. Therefore, other biocontrol agents may be necessary to target the exposed areas of the crop or crops without a closed canopy. Another factor to consider is that mites cannot fly, so they require a bridge of plant material to travel between plants. The temperature range and relative humidity provided in this table are a guide only, and they should always be discussed with the biocontrol providers. Some biopesticides, such as bacteria or viruses, must be consumed by the insect in order to kill it. These control methods can be applied for insects feeding on the crop, but the timing of application is essential here, if only certain life stages of the insect are consuming the crop, especially because these biopesticides are not persistent. For example, several biopesticides based on baculoviruses and the bacterium Bacillus thuringiensis can be used to target Lepidopteran caterpillars. The codling moth can be effectively controlled by codling moth granular virus. Careful monitoring of the crop is required to time applications of the virus onto the leaves so that caterpillars pick up a lethal dose of the virus after they emerge from the eggs laid on the leaf, but before they bore into the apple. If you are targeting Hemiptera or Homoptera, such as aphids, whitefly and plant hoppers, biopesticides that must be consumed will not work, as these pests have piercing, sucking mouthparts. They feed on the phloem of the plant using a straw-like structure, and if you have applied a virus or bacteria to the plant leaves, the insect will not consume it. Instead, we can use contact-acting biopesticides such as fungi. These can often be applied by similar methods to chemical pesticides, but we need to consider whether certain life stages of the pest will be more susceptible to the biopesticide and think about how we can target the pest. For example, aphids and whitefly are often found on the underside of leaves, and so for successful applications of entopathogenic fungi, the biopesticide should be applied using a sprayer which targets the underside of leaves. Other species of invertebrate also play a vital role in agroecosystems. Butterflies, moths, bees, flies and beetles help pollinate our crops. Dung beetles play a vital role in the nutrient cycling of dung in livestock and mixed farming systems. And these are just a few examples of the range of invertebrates that benefit our cropping systems. It's important to consider and encourage all beneficial invertebrates for a holistic approach to IPM. To summarise, there are many biocontrol technologies available that can be used as part of IPM. Selection of suitable biocontrol options is dependent on the crop, environmental conditions, and the likely pest problems. For successful biological control, it's essential to use the right species in the right number at the right time. The IPM strategy needs to be carefully planned, timed, and managed, and other IPM tactics selected for their compatibility with the biocontrols used. If you would like to read more about the references and resources used to develop this chapter, please pause here.